Welcome back, everyone. So we're going to continue on learning about cellular respiration in chapter 5 with how cells obtain energy. So in the previous video we learned about glycolysis, which occurs outside of the mitochondrium in the cytoplasm. So we're going to continue with the products of glycolysis, pyruvate, moving into the mitochondrium and continuing to be broken down further through the citric acid cycle and oxidative phosphorylation to extract ATP. So that's what we'll continue to look at. So by the end of oxidative phosphorylation, we will have extracted enough energy to basically get about anywhere from 32 to 36 ATP from that one single glucose molecule. So by the end of this video, you will be able to describe the location of the citric acid cycle and oxidative phosphorylation in the cell. You should also be able to describe the overall outcome of the citric acid cycle and oxidative phosphorylation in terms of the products of each, as well as describe the relationships of glycolysis, the citric acid cycle, and oxidative phosphorylation in terms of their inputs and outputs. So just like in the previous video, that chart that I suggested that you created, we'll continue to add to that. So in the citric acid cycle, we'll look at the inputs and the outputs and how much ATP was generated, as well as in the electron transport chain and oxidative phosphorylation, how much was produced there. And then there's also an intermediate step where we'll talk about that as well. Okay? So just to begin with, let's take a look at where we left off. So with glycolysis, remember with glycolysis, this is occurring outside of the mitochondrium in the cytoplasm of the cell. And this is something that all cells have some form of glycolysis. So we're extracting energy from glucose. So we're taking that glucose molecule and essentially we're just breaking it, that 6-carbon molecule, into 2 3-carbon chains of pyruvate. And by doing so, we're extracting some energy, right, in the form of ATP. So by doing that, we get 2 ATP from that process, as well as 2 of the electrons are stripped from glucose and are added to our electron carriers, NADH. Okay, so you can see those electron carriers are going to be shuttled into the mitochondrium where they'll be used later on in the electron transport chain. We'll get to that in a little bit. So next we're going to talk about this pyruvate. So the two 3-carbon chain pyruvate molecules are going to now travel into the mitochondrium. And there they're going to undergo the citric acid cycle as well as the electron transport chain. Okay, and we will see how they're going to extract even more energy from these processes. And at the end, we're going to end up with 36 ATP extracted from that single molecule of glucose. Okay. So let's begin with what we call the transition step. So it's not the citric acid cycle, it's not the electron transport chain / oxidative phosphorylation. So in your chart, you can just write it as the transition step. Okay, because nothing is really no ATP is being produced, I should say. Okay, so in this case, what we're doing is we're essentially just doing a minor step to prepare for the citric acid cycle. So this does occur in the mitochondrium. Okay, so the pyruvate produced in glycolysis is transported into the mitochondrium. So here's our pyruvate, our pyruvic acid. And you can see what is taking place here in this reaction. So in this reaction, one of the carbons is going to be stripped off, okay, to form carbon dioxide. So that's going to leave. And then the 2-carbon chain is now going to get added to a coenzyme. And you remember from the previous last week when we talked about coenzymes, they're kind of like, helpers to enzymes. So this coenzyme, it's called coenzyme A, is going to be added to the 2-carbon chain to form something called acetyl-CoA. Okay, so we have acetyl-CoA being formed, and in that process, when we're breaking that carbon off from pyruvate, we're actually going to strip a couple electrons off of that as well to form an NADH. Okay, so the end product of this process, we end up with acetyl-CoA, a carbon dioxide, and an NADH. Okay, and this is happening twice, right? Because we have 2 pyruvic acids or 2 pyruvates. So we end up with a total of 2 acetyl-CoA, 2 NADH, and 2 carbon dioxides. Okay, so those will be entering the citric acid cycle. Okay, so here are the inputs and outputs of the process. So again, we have our 2 3-carbon pyruvates that are entering the mitochondrium, and those will go through this transition step where we're going to end up with 2 2-carbon acetyl-CoAs. So they have 2 carbons with the coenzyme A attached. We also, our input is coenzyme A, okay, but because that got added to our 2-carbon chain, we lost that carbon dioxide, so we have 2 carbon dioxides that are going to leave. And then we have a couple of electrons that were stripped in the process, so they're going to be added on to NAD+. So NAD+ will get reduced to NADH. So those are the outputs. All right, and of course the NADH will then travel towards the electron transport chain where those electrons will be used in the electron transport chain later on. All right. All right, so now we're going to move on to the actual, next step, the citric acid cycle. So this citric acid cycle will produce ATP, so this will, be an actual step in the process of cellular respiration. And sometimes you will see it as the Krebs cycle. We typically don't use that term Krebs cycle anymore, mainly because originally it was named after this scientist named Krebs, who was a contributing member, a scientist who helped discover the process. But he wasn't the only one involved, so that's why, why should he get the credit when he wasn't the only one involved? So, a lot of scientists decided that, maybe we should just call it the citric acid cycle instead of just naming it after one person when multiple people were involved in the discovery. And so if you see it as Krebs cycle, just realize it's the same as the citric acid cycle. So this is going to occur in the matrix of the mitochondrium. So let me see if I have a picture of, here's our mitochondrium. Remember, the mitochondrium is like this little kidney bean shaped thing. And if you recall way back in, when we were talking about the structure of the mitochondrium in chapter 3, we learned that the mitochondrium has a double membrane. And so here is that double membrane. And these little folds, these are the inner membrane, And inside the inner membrane, okay, so we have the outer membrane, the inner membrane, and then right here, this is called the matrix. So like where this word is, mitochondrium, this is called the mitochondrial matrix. And so the citric acid cycle actually takes place in the mitochondrial matrix of the cell, of the mitochondria. Okay, so it occurs in the matrix of the mitochondrium, so that inner space of the mitochondrium. And if you can see here, it is a cycle. Okay, so it's a closed loop, and it's a series of 8 chemical reactions. And luckily you don't have to remember all the products of each of the reactions. You just need to know the beginning and the ending. So let's look at the beginning. Oops, let me go back. So we're going to begin with the acetyl-CoA. Okay, so this 2-carbon acetyl-CoA is going to enter the citric acid cycle. And as it's entering the citric acid cycle, the coenzyme A will break off and get reused. Okay, so that's just going to go back over here and attach to another pyruvate. Okay, so we're only entering the citric acid cycle with this 2-carbon chain. So that 2-carbon chain will actually get added to a 4-carbon chain that's already in here. And then that 4-carbon chain will get added to the 2-carbon chain to form the 6-carbon chain. And then that'll get reduced and reduced and reduced and reduced and reduced and reduced until we end up with just this 4-carbon chain again. And so we end up losing two carbon dioxides in the process. Okay, so there's where the 2 carbons end up. They're going to be leaving the cell. And by this point, you'll notice that we've lost all of the carbons from the original glucose molecule. Okay, so we lost 2 up here. We lost another 2 here. Okay, or we lost 1 here, we lost 2 here, and then this is happening twice because we have 2 pyruvate, so we lost a total of 6 total. So our glucose molecule, by the end of the citric acid cycle, has been completely destroyed. All the carbons have been gone, okay? All the oxygens from that have been gone too, okay? In that process of like breaking down and moving through the citric acid cycle, and it is complicated, and so we're not going to memorize all the little reactions, that all the 8 steps that occur. Just realize that we end up losing the carbons to carbon dioxide, and we end up reducing, those electrons get stripped and added to NADH, and another electron carrier called FAD to FADH2. So we have 3 NAD plus that will get their electrons to reduce to 3 NADH. So those will get shuttled to the electron transport chain. We will also have FAD, another electron carrier, get reduced to FADH2, and FADH2 will carry those electrons to the electron transport chain. Okay, so in the end, we have a bunch of electron carriers shuttled to the electron transport chain. Okay, we also gain some ATP from this process. So we gain 1 ATP for every acetyl-CoA that went in there. All right. So this is the total product per acetyl-CoA. Okay, and remember we have 2 of them that go in there. We have 2 pyruvates that went into the mitochondrium. So this, citric acid cycle is going twice. So we end up with this times 2. Okay. And so by the end of this whole process, we have completely broken down glucose, okay, from our original reactants. We haven't even touched oxygen yet from cellular respiration. We'll get to that later. So we've completely broken down that glucose, and we've extracted a little bit of energy from that process. And a lot of electron shuttles have been shuttling electrons toward the electron transport chain. So here's the summary of our citric acid cycle. So our inputs, we have 2 2-carbon acetyl-CoAs from that transition step. Okay, so those will enter the citric acid cycle. The coenzyme A will leave to get reused, and then this 2 2-carbon carbon chain will then leave the citric acid cycle as 2 carbon dioxides. We have 6 NAD plus that will get reduced to 6 NADH from the electrons that get added to it. We have 2 FAD that will get reduced to 2 FADH2s. And then we will get some energy, so we have 2 ADP that will end up getting phosphates added to them to produce 2 ATP. So we do end up with some energy there. Okay. All right, so back to our equation. We've completely gotten rid of our glucose from that process. We started here in glycolysis. We broke it down a little bit in glycolysis in the transition step and the citric acid cycle. Now let's figure out what's going on with this oxygen part. Okay, so we're going to move into this last step. So let's talk about the electron transport chain. So remember all of those electron carriers, the NADH and the FADH2s that are carrying electrons from glycolysis, from citric acid cycle, from that transition step. Well, they're all heading towards the inner membrane of the mitochondrium. So again, let's go back. Remember, we have the mitochondrial matrix right here. The electron transport chain will take place in the intermembranes right here. Okay, so this is the outer membrane, this is the inner membrane, this is the mitochondrial matrix, and then this is the inner membrane space. So we're going to be talking about this inner membrane, and this is where the electron transport chain will take place. Okay. So here is that inner membrane. And again, here's that mitochondrial matrix, and here's the inner membrane space. So you can see how the electron transport chain involves, basically we have these transmembrane proteins, okay, see, and peripheral proteins, you can see them embedded within the inner membrane. And you can see how the NADH and the FADH2 are going to be oxidized. Remember, oxidized oil rig, or oxidation is losing electrons. They're going to be oxidized or lose their electrons. So the electrons are going to be carried through the electron transport chain. So those electrons pass through this complex of proteins in the inner membrane. And as they're passing through, you can see that the NADH will be oxidized to NAD+, and then that just gets recycled back out into the cytoplasm where it can accept more electrons. And FADH2 will get oxidized to FAD, where then it can then go back to the citric acid cycle and accept more electrons and get reused. Okay? So as those electrons are moving through, they will finally end up getting accepted by oxygen at the end. So oxygen will finally come into play here as the final electron acceptor. So oxygen will receive those extra electrons at the end. Okay. When those electrons pass through, they're going to end up opening up these channels. They're going to create this, what's called an electrochemical gradient. And as they create this electrochemical gradient, it's going to allow for basically protons to be pumped across the membrane into the inner membrane space. Okay. So these electrons open up these channels here. You can see this channel, this channel, this channel. And a flood of protons get pumped across the membrane into this inner membrane space. So now we have all of these protons on this side of the membrane. And this is creating an electrochemical gradient in this inner membrane space. Right? So once we have all of these protons on this side of the membrane, we're going to end up with this enzyme being activated. So this is a special enzyme which we call ATP synthase. And remember, synthase, ACE, anything that ends in an ASE is some sort of enzyme. So this is a special enzyme. And when this enzyme starts functioning, it's going to open up, and the basically all of these hydrogens are going to flow down their electrochemical gradient through the ATP synthase enzyme, and a little propeller is going to form in here, and the flow is going to turn these little generators that generate ATP from a bunch of ADPs. So it's just gonna sit there and provide all this energy to form ATP from ADPs. All right. So as this is forming, we're going to have all these hydrogens flow into the mitochondrial matrix. Okay. So as this is happening, all of these hydrogens will get added into the mitochondrial space, and they can get added to the oxygen, that's the final electronic acceptor, to form water. Okay. So this is what we call chemiosmosis. So chemiosmosis is basically just chemical osmosis. So basically the flood of hydrogens across the membrane. And this whole process is a high yielding energy source. So this is yielding anywhere from 30 to 32 ATP during this process. All right. Now that's just from this chemiosmosis part. So add that 30 to 32 ATP plus what we earned from the citric acid cycle plus what we earned from glycolysis. That's where we add into, anywhere we end up with about 36 ATP. All right. Okay, so here's our summary. So from glycolysis and the transition step and citric acid cycle, we had a total of 10 NADH enter the electron transport chain. And so those 10 NADH were stripped of their electrons, and so they were oxidized 10 NAD+. We had those 2 NADH2s from the citric acid cycle that were stripped of their electrons to form 2 FAD. We had 6 oxygens that were brought in, originally from breathing in from her lungs. They were the final electron acceptors to form water. And then we had about 30 ADP plus the phosphates to form about 30 ATP. All right. So there's our output. So we end up, you can see that this portion of cellular respiration yields the highest amount of ATP. So here's the payoff phase. We end up with the most. All right. So basically here's the whole process, by the way. So you can see the citric acid cycle is taking place in the matrix. And then all those electron carriers, travel to the electron transport chain where their, electrons get transported through the electron transport chain, opening up the proton pumps, channeling the protons into the inner membrane space. Okay, and then the ATP synthase is propelling those hydrogens back into the matrix, using that energy to make the ATP, right? So here is a question from your textbook. So this is an art connection question. Cyanide. So you've probably heard of cyanide. It's a type of poison. It inhibits something called cytochrome C oxidase. So it's a component of the electron transport chain. So if cyanide poisoning occurs, would you expect the pH of the inner membrane space to increase or decrease? What effect would cyanide have on ATP synthase? So if cyanide poisoning occurs, so cyanide inhibits cytochrome C oxidase, a component of the electron transport chain. So if cytochrome C is a component of the electron transport chain, and cyanide inhibits it, imagine something's happening here, shuts it down. If this gets shut down, what would happen to the pH of this inner membrane space? Do you think it would increase or decrease? If you said increase, I would agree with you. Okay, the reason why it would increase is because would we have hydrogens being pumped through? No. Okay, because this would be shut down, right? If this is shut down, then we wouldn't have hydrogens pumped through. If we don't have hydrogens pumped through, hydrogen ions pumped through, then the pH would not go down. And so the pH would increase. Because remember, lower pHs mean higher hydrogen ion concentration. So we don't have hydrogen ion concentration, being concentrated up here. So we have an increase in pH. Okay. What would cyanide have on ATP synthesis? What would happen here? It would stop, right? It would be inhibited, because if this shuts down, so would this. Okay, so this would inhibit ATP synthesis. So you can think of like cyanide poisoning, it's basically going to cut down this whole process, and so we're not going to be making ATP. And so this is going to cause, the body to shut down. All right, so here is our whole process from start to finish. So we started out with glucose that was brought in. It went through glycolysis, where it broke down into pyruvate, 2 pyruvate molecules. Those pyruvate, and then moved into the mitochondrial matrix, where they entered that transition step, where they were stripped of a carbon to form acetyl-CoA. That entered the citric acid cycle, where they were completely broken down into carbon dioxide. And then the electron carriers were traveled to the electron transport chain, where they finally were accepted by the oxygen. And then that was used to power the ATP synthase in order to generate ATP. And so let's see if we can review with some review questions. So what do the electrons added to NAD+ do? Is it A, they become part of a fermentation pathway, B, they go to another pathway for ATP production, C, they energize the entry of the acetyl group into the citric acid cycle, or D, they are converted into NADP. So what is the purpose of the electrons added to NAD+? If you chose B, I would agree with you. So remember, when we are basically reducing NAD+ to NADH, the NADH is an electron shuttle, and the electrons then travel to the electron transport chain, where they're going to be used for ATP production. So B is the correct answer. Here's another question. Chemiosmosis involves A, the movement of electrons across the cell membrane, B, the movement of hydrogen atoms across a mitochondrial membrane, C, the movement of hydrogen ions across a mitochondrial membrane, or D, the movement of glucose through the cell membrane? So what does chemoosmosis involve? If you chose C, I would agree with you. So remember, in chemoosmosis, we're not talking about the electrons moving across the cell membrane. We're talking about the hydrogen ions moving across the cell membrane. Okay, the electrons are moving through the membrane. Okay, the hydrogen ions are moving across from the matrix into the inner membrane space. Okay, so the movement of hydrogen ions across the mitochondrial membrane. Okay, so C is the correct answer. And then finally we have, in cellular respiration, glucose is broken down to produce the end products. Is it A, NADH, water and oxygen, B, NADH, FADH2 and water? Is it C, ATP energy, water and carbon dioxide? Or is it D, ATP energy, water and oxygen? So what is glucose broken down to produce? If you chose C, I would agree with you. So remember, glucose is broken down during glycolysis as well as in the citric acid cycle. And the end products of those is carbon dioxide. And then we're using those electrons and eventually to produce water. And then we have ATP energy produced. So glucose will end up becoming carbon dioxide and water. All right? And then the energy, ATP. So C is the correct answer. Okay, so that is it for 5.2, the citric acid cycle and oxidative phosphorylation. So by now you should be able to describe the location of the citric acid cycle as well as oxidative phosphorylation in the cell. And you should be able to describe the overall outcome of the citric acid cycle as well as oxidative phosphorylation in terms of the products of each. And then finally, you should be able to describe the relationships of glycolysis, the citric acid cycle, and oxidative phosphorylation in terms of their inputs and outputs. Okay? So in our next video, we're going to take a look at what happens if we don't have oxygen present. Okay? So the video that we just watched talked a little bit about what happens when oxygen's not present, how we can still extract energy using glycolysis through a process known as fermentation. So we'll talk about fermentation in the next video.